get a thumbs up, I'll go.
Oliver, yes, you are live.
Oh, okay.
Go, go, go. Every time, every time.
Welcome, everybody, to yet another Journal Club. Welcome to the holiday season. I'm Oliver Medvedik, broadcasting, I guess, somewhat live, from New York City, the Kambar Center for Biomedical Research over here at Cooper Union. And we have yet another Journal Club to present to you on behalf of LEAF Life Extension Advocacy Foundation and Lifespan.io. Just going to do one more shout out since it is the holidays and it is the time for giving, that we are having a campaign right now for our recurring donors. So those of you who are enjoying knowledge that we're dropping here, feel free to donate. Whatever you can spare, $1, more, less, whatever. You can go on our website, livestreamed.io, and be a recurring donor, a lifespan hero, as we call them. And I am also a lifespan hero, so I am donating to myself. I'm not exactly how that really works, but I do that as well. So welcome, everybody. And this paper that we have here, I am really, really, really psyched about. It is a paper about a senolytic. I know we've talked about senolytics out the years, but what I really like about this paper is that it deals with a compound that we've heard of and we mentioned on our last broadcast, which is Fisetin. It's a plant flavonoid, so these multi-grain compounds that give fruits and vegetables their colors, and a lot of them have positive health implications. And Fisetin is one of these compounds that's known as a senolytic or senomorphic, or seno something that improves basically the clearance of senescent cells in vivo and in vitro. And Fisetin, of pretty much all or most of the senolytics that have been studied, has an outstanding safety profile. I can pull up references later. I don't have them right now. We're going to focus on the paper that we discussed, but I mean, you can literally feed like grams per kilogram, to mice, and there is no acute toxicity. And people have been taking it as a supplement. There are supplements out there, and short of looking at pharmacokinetics and bioavailability, which is something that still needs to be done for humans, the safety profile, as far as I'm concerned, is pretty rock solid. And the paper that we're going to be looking at here deals with an animal trial, both in vitro using cells, both human cells and mouse cells, and also mice that are both an accelerated aging phenotype, as well as normally aged mice. And again, the results are pretty impressive. So, I'm going to kind of bookmark this entire presentation by saying, why aren't there more aggressive clinical trials with Fisetin out there? If you go on to at least the US clinical trial database here in the United States, there's maybe a handful, maybe 2 clinical trials using very modest piddly doses of Fisetin. I'm not sure why, since it's a senolytic, so you really need to take a high enough dose to initiate senescence clearance. But that being said, the other positive aspect about a senolytic such as Fisetin, besides being bioavailable in the right formulation, incredibly safe, little or no toxicity that's been identified in mammals that are tested. But also that it's not something you need to chronically take, right? So if it's working the way it's supposed to be working, which is as a senolytic or senomorphic, we're talking about an acute dose that's delivered within a time frame of, like, for at least mice that they did the study, days or weeks, right? And the other really amazing thing about this paper is that they achieved very positive effects in mice that were, the equivalent of a 75-year-old. So this was delivered to mice that were basically about 20 to 24 months of age, which is pretty darn old for a mouse. And they, achieved substantial gains in lifespan, both mean and maximal lifespan. So that's sort of the gold standard for longevity studies. So, I guess with all that said, let's get right down to it. Let's share the paper here. Screen. Click on that. There we go. And voila. Okay, is that appearing very clear to everybody? You can see the title, more or less. Good.
Looks okay to me. If people are struggling, there is a link in the description of the show anyway for them to get the paper. It is an open access paper, so if people want to read along with a full-size copy, or perhaps have got the luxury of a second screen, they can do. But yeah, the link's in the description.
So yeah, so this is one, so another reason why I'm excited by Fisetin, if all of those reasons I haven't given you are sufficient, is that it's a very well-studied molecule at this point. It's been looked at in many labs. So we're not just talking about one study that's just, come out of one lab, but many, many labs over many, many years for many indications, including, Alzheimer's mouse models, various cancers, so on and so forth. So, I would think with all of this data that it would be very aggressively being pursued as the compound to, look at its pharmacokinetics to make sure that the blood delivery is there and get right into many human clinical trials. And that's something that we here at Leaf are actually something that I'm doing some research on, is to see if we can set up eventually some crowdsourced trials to see the effects. Okay, so the paper itself is Fisetin is a senotherapeutic that extends health and lifespan. I like how they use the term senotherapeutic because we're not 100% sure if it's clearing the senescent cells through apoptosis, or they're xenomorphic, they're changing back to normal, or exactly how they're being cleared. There's a number of collaborators here, so it's coming also from the Mayo Clinic and the Scripps Research Institute. So a lot of top labs. So let's take a look and see what sort of data that they have here. And this paper came out pretty recently. It's 2018. I believe it came out in October. Actually, here's the history: received July, revised, etc., finally accepted September. So pretty recent paper. So let's scroll through here and let's basically see what they look at. Scroll, results, right to the first figure. Okay, so what they first did was they looked at a panel of different types of senolytics, or different types of compounds that perhaps get reversed, not senolytics, xenotherapeutics, things that can clear senescent cells. They used, I believe they used a kind of a, here they used beta-galactosidase staining. That's a kind of a marker for senescent cells. They, counted total cells, so relative cell number, so total cells and the percentage senescent cells. And they used a 5 micromolar, they said. So it's basically a screen. And what they used for the screen were these ERCC1 double negative MAFs, murine embryonic fibroblasts. So basically a mouse cells that are defective for ERCC1, which is required for basically DNA repair. And I believe these were passage 5, so they're passage multiple times. And I think they were also grown in a higher oxygen environment. That might be another experiment. So essentially, this was inducing senescence. And they looked at compounds that basically, were already studied before, and probably a whole bunch of others. And they wanted to pick the ones that had the best clearance or reversion of senescent cells by, using this drop in beta-gal levels. And Fisetin, you can see here, had the most dramatic response. Curcumin is kind of the second runner-up, which is the active ingredient in turmeric. So a lot of you, know these spices that have beneficial effects, and a whole bunch of others. Interestingly, quercetin didn't really have an effect, which is, it'd be interesting to revisit some of these because maybe this was maybe a very high stringency type of assay. But suffice it to say, Fisetin did really well, and it's got an excellent safety profile. So they decided to run with Fisetin. That's not to say that probably some of these as well couldn't be revisited in this, in these assays. B basically shows, over increasing doses what happens. So basically, just like figure 1A, figure 1B is over increasing dosage from, looks like 1 micromolar, you're starting to see a dramatic effect down to 20 micromolar. So you see, the relative amount of senescent cells in the population go down in these ERCC1 double negative murine embryonic fibroblasts. And they do the same thing with IMR90 cells. So these are, I believe, a human tumor cell line that was treated with this chemotherapeutic called etoposide, and that induces senescence. And adding Fisetin, so here they have increasing doses of Fisetin, 7.5 to 15 micromolar. It's also dropping, the amount of senescent cells per this assay that they used with beta-galactosidase expression, which is a typical assay. So, kind of nice, simple data that in their initial screen, and again, the data gets better and better through this paper because then they start looking at, mice, but as an initial screen, it's like, okay, so if you see this data, you're like, all right, well, a lot more work needs to be done because, we're talking about mutant cells in dishes, and okay, it's got some effect, but, how does it really behave when you look at the actual animal? And that's what they really do later. So now we start getting into some really interesting stuff. They now are looking at animal models, and they have these mice here that have this novel, it's a really neat reporter where basically, so P16 is a tumor suppressor and it's upregulated in senescent cells. So you can use the promoter for P16 to basically switch on all sorts of reporter genes. So reporter gene is just something that, tells you the functionality of something else, in this case, the P16 promoter. So that's essentially your proxy for senescent cells, right? And what the P16, what's happening here is as cells senesce, this promoter gets turned on, and that turns on the expression of a reporter gene, which is Luciferase. And Luciferase is a gene that basically, an enzyme that causes cells to essentially glow in the dark if you add in a compound that they oxidize. So it's a very typical reporter, right? And you can then, these are whole bodies of mice that are put under a machine that can detect photon emission, luminescence. And these are done in these ERCC1 minus delta. And basically what that is these cells are partially, well, they're missing ERCC1, and then they have a defective, another kind of slight defective copy of ERCC1, because I think having a double minus for a mouse would be pretty lethal and they wouldn't be able to grow very much. So they use these really weakened mice that are for ERCC1, which they state are progeroid mice. So they have, DNA repair defects. So they accumulate senescent cells basically pretty quickly. And you can see here with control and Fisetin, and this is the protocol that they use, a control diet, Fisetin diet. They said they were fed a diet. So they use different diets throughout this paper. They either administer it in their food, the Fisetin, or they give it through oral gavage, which means you essentially ram a syringe down a mouse's throat. That's basically what it looks like. And you squirt the Fisetin with a carrier, a slurry, if you will, of something that can dissolve it into the mouse's stomach. And, here they say it's 500 mg per kg. So that's a very high dose, 500, so half a gram per kilogram of Fisetin, but that's given throughout the duration of these 2-week periods. So you can go into the materials and methods and kind of work that out for yourself, how that's run. But these are very, temporary administration. So Fisetin, so here are age in weeks. So these mice don't live very long. I don't know what the maximal lifespan is exactly. But mice starting at 4 weeks all the way to 16 weeks, control diet in the black, Fisetin diet in the red. And, and they were given like 2-week administration. So week 6.1 to 8, and then 12.1 to 14. So you can already see within the period of administration, after 2 weeks, you can see already a significant drop in luminescence. Here's, I guess, an example. Let's see, representative age range. So 12-week-old male. So right around this point is Fisetin in control, and you can see significantly less, photons being emitted from, which is not really sure where you have the concentration of these cells. I don't know if that's adipose tissue or, it'd be interesting to see if you can kind of if there's a way to really find out where most of the senescent cells are being cleared. I'd be interested by tissue, but they do that later with when they sacrifice the animals. But it'd be interesting if that could be applied into this, this whole body scan. But you get this dramatic reduction, right? So it's significant, and increase in luminescence goes up with control diet and stays much more lower significantly in these progeroid mice over time. So again, these were not fed through, they weren't feeding these mice continuously. They were basically fed at once, and these levels, during this time seem to lag, even after the Fisetin was removed, and then they, for some reason, dosed the mice again with Fisetin, although I'm not sure if that was really necessary, now that we sort of understand how the concept of senolytics works. Okay, I'm going to stop sharing for a little while. So I just went through 2 figures, so we got a couple more. So let's see, question. Do people already occasionally take senolytics with their food, strawberries, apples, berries, so easily? I mean, children lives 0 senolytics, right? We humans have already get curcumin by Z. Okay, so I'm going to read that more slowly. Okay, there's a question. Since people already occasionally take senolytics with their food, strawberries, apples, or curry, can we still expect to translate the results from mice to humans so easily? Control group in the mice experiment probably got 0 senolytics, right? Probably. I don't know what's in mouse chow, so it's probably not 0. I mean, if it's a ground up, basically vegetative, matter and other stuff, so it's probably not 0. It's whatever the mouse chow is, and you can certainly look up the ingredients. We humans already get curcumin and Fisetin from food, and maybe others. Well, depending on who you are, right? A lot of people eat a lot of garbage, and I could say that probably for a lot of people, curcumin and Fisetin levels are pretty low if you're not eating fresh fruits and vegetables every day. And also, you have to realize that the amount of Fisetin in strawberries, which is actually at the high end, it's pretty low. There's a paper somewhere. It's something around the micrograms per kilogram, and we're talking about here. They're giving milligrams of this stuff. So even in food that's loaded with flavonoids, it's still relatively pretty low compared to what experimentally is being administered to have a senolytic activity. Now, one caveat I'll mention is for people wanting to replicate any of this. First off, you can't go directly from a mouse weight to human weight and just linearly extrapolate, right? So according to that extrapolation, if you went from, a mouse taking, it was somewhere here between 20 to 100 milligrams per kilogram, we're talking well over a gram of Fisetin you'd have to eat. That's, there's lots of adjustments that have to be made, and I'm definitely not an expert on this, but I've read some papers in preparation for this where you have to do something called body surface area scaling, so there's a factor for that, but that only applies to things that have a toxic dose level, and Fisetin doesn't appear to have any toxicity, so you can probably safely go much higher than what's recommended as a starting dose if you're just going to look at let's say your typical phase 1 trial for toxicity profiling. That being said, certain compounds get absorbed differently between mouse guts and human guts. So, one thing that really needs to be done, and I think should be done immediately, if we don't have the data on it, is pharmacokinetics in humans. The other issue is delivery. You have to realize that they mix this. Fisetin here was also administered to the mice, both mixed in food and also oral gavage that was mixed with a number of carriers like polyethylene glycol, which basically, probably makes absorption a lot better than if you were to just take a dry compound. And I know there's a lot of studies out there with other compounds such as curcumin, where, you know, just taking it as a pill form is not as effective as if you have it mixed with some sort of carrier. So, you know, that's something to really, you know, if you want to make sure that the trial is done right, so you don't basically dismiss something that's supposedly not working, but the reason it's not working is it's just not even getting into the bloodstream. You have to make sure that you look at the pharmacokinetics, which means that you have to take a look at, is it actually being delivered to the blood, right, after you've eaten the stuff. So, so I guess that's a very long answer to Marcus, I think, Buechner's question. It's, you know, it's not clear, and the amount of Fisetin you're taking from food from the get-go is pretty low for senolytic activity. Let's see, Tony Fatica. Can C-reactive protein from blood work be an effective measurement tool to determine if Fisetin help reduce inflammation? If Fisetin is sending senescent cells into apoptosis and reducing toxic cytokines, I presume it will reduce inflammation and see results from this measure. I guess, I don't know. I don't want to comment on this because I don't know what would be a proxy measure. I'd have to do a little bit more reading, you know, to see what would be an effective proxy measure. I honestly don't know, and I'm sure that there's studies out there that we can then pull up later, maybe during our fireside chat, to see, you know, what would be good blood measurements to take for effectiveness.
Well, there is a problem, and the paper actually mentions that problem as well, and that is that Fisetin inhibits NF-kappa B signaling. Mm-hmm. So it works as an anti-inflammatory agent to a completely independent pathway. So in that sense, you will always see probably a decrease in inflammation, mm-hmm. but that doesn't reflect anything to the senolytic effect. Mm-hmm.
Yeah, a lot of these flavonoid compounds have multiple targets. If you just pull up a paper, if you want to find out, gee, what does Fisetin actually bind to? I don't know. There's a lot of there appears to be a lot of targets. So it's not exactly clear. The downstream effects are clearer than a lot of the upstream kind of targeting effects. And Steve, you said that 100 milligrams is a typical supplement dose?
Yeah, at a rough look. A lot of these flavonoids tend to be chemically quite similar. I'm interested to see how similar Fisetin is in comparison to, say, quercetin or apigenin. We touched upon this last week, didn't we?
Pretty similar. We're talking about they're off by like a couple, like one or two hydroxyl groups. I mean, flavonoids.
Actually, it's one hydroxyl group different. I looked it up.
Yeah, which is what, as I mentioned before, which is why I think apigenin, which chamomile, I think that's possibly another candidate. I'd like to get that tested as well. To circle back to what you said earlier about why aren't we doing these you know, these trials? They're not clinical trials because they're generally regarded as safe, but you know, these things should be relatively cheap to do.
Yeah.
And I think it's low hanging fruit, isn't it?
No pun intended. Definitely is low hanging Fisetin enriched fruit. Yeah.
Low hanging strawberries or whatever.
One gigantic strawberry. Okay, so let's. Excellent questions. One I can answer somewhat, kind of. The other one, I don't want to jump into an answer because I don't know what would be a good proxy measure in the blood. Share screen. Okay. All right. Back to our results. All right. So next up is basically, well, they're not biopsies. Actually, I believe sacrificed these mice and just took samples of cells, and they looked at the expression of senescence-associated factors that are correlate with SASP, right? Senescence-associated secretory phenotype. So they looked at tissues from 20-week-old male and female ERCC, minus delta mice that were chronically exposed to Fisetin through their diet or fed a control diet. And they looked at various tissues, so fat, spleen, liver, kidney, CD3+, which I believe are T cells. And they looked at, I believe these were from in liver. They looked at these hydroxy non-enol adducts, which are basically lipid. I believe it's a lipid oxidation, or basically sort of malformed lipids in the plasma membrane, which are indicative of stress, and then a ratio of reduced glutathione, right, versus oxidized glutathione. So the higher, the more reduced. That means the liver is not being as stressed because glutathione is basically used as a compound within cells to detoxify, right? So just looking at this, you could see a difference just kind of in one glance, right? So this is a panel P16, P21, so your tumor suppressors, a whole bunch of other factors associated with SASP. ERCC1 minus delta is in red, and you can see that there are drops in basically all of these factors in fat, in spleen, in kidney, relative expression level. So quite significant for quite a few, right? It looks like at least 50% for quite a number of them. And these are just the same profile. So, I'm not going to hammer each of these individually, but, that's a snapshot. So Fisetin is basically, if you look at, if you basically, do qRT-PCR, basically look at RNA levels of all of these factors that are associated with SASP, they go down dramatically throughout a whole bunch of tissues. And these are from, again, 20-week-old mice from the treatment that was seen in the prior figure, right? So pretty good. Next up, they do acute Fisetin treatment. So this is basically treatment that's much shorter duration. And what's cool about this one is they were looking at, okay, so these are aged wild type mice and also human explants. So down here, E and F are human explants. So I'm trying to wrote this down in my notes here, where exactly which human cells were used. So now we start getting into, human adipose. Yeah, adipose tissue explants. And so these are old wild type mice, but I know that they used a reporter gene here, and so it's so the wild type C57 black 6 mice were given Fisetin 100 mg per kg or vehicle for 5 days by oral gavage, right? So basically, so 100 mg per kg, I believe what that means is that they were given across the 5 days, so basically 20 mg per kilogram each day by oral gavage. And if you can look in the materials and methods, it's like a slurry with a couple of different compounds, 10% ethanol. So it's not just like powdered Fisetin. And then 72 hours of the final dose, the mice were sacrificed, and the number of beta-gal cells, expressing cells, were quantified in fat. So this is figure A, right? And you can see that the number is percentage, so percentage of all nuclei relative to vehicle, it's quite a big difference, right? So this is just in wild type cells. What they do next is they have these transgenic cells, are these mice, or yeah, these ink attack mice. So what that is it's basically a reporter that's tied to P16 again. And these are transgenic mice that express this. And normally what this allows the researchers to do is to ablate, I believe, senescent cells. There's another, there's a fat attack mouse, which basically uses a fat promoter, so you can ablate adipose tissue. And this is a modification of this called ink attack. So because P16 is also known as A4A. They don't use this to ablate the cells. What they use this is basically as a reporter in all tissues. So basically you can then look at cells that are expressing this, right? If they're expressing this, it's sort of like the marker for beta-gal that's used. you can measure GFP levels, and I think they use an antibody against this CASP-A flag, which can be switched on. But I know it's a little complicated here, but they're basically using this as a reporter to tag various cells that are turning on anything with a P16 promoter, i.e. senescent cells. So P16 is just used as this proxy. And I'm trying to look here and see. So these are aged ink attack male mice. So these are aged mice, pretty old. So even though they're transgenic, they live fairly normally, I believe. So age 22 to 24 months. These are acutely treated also with Fisetin. So these are old mice treated as above. So acute dosage, right? 5 days oral gavage. And then they looked at these flag tags. So they looked at the expression of this reporter in various tissues, right? So, you know, the open bars are young mice as controls. I think young mice, how old are the young mice here? What's young for a young mouse? 6-month-old, I believe, right? So 6 months versus 24. Old is, you know, 2 years or so. And then old, this is, we filled in with black is old plus vehicle, and then old plus Fisetin. So c-KIT, I believe, is a marker for, so these are mesenchymal stem cells, CD4 positive T cells, CD8 positive T cells, so helper T cells, cytotoxic T cells, natural killer cells, endothelial cells. They also use another marker for senescing cells here, other than, I believe, instead of using this flag tag, they use the SEN-VP, which is an antibody against centromeres that I believe the centromeres get less compacted. So that's another marker for senescence that they use. And they look at c-KIT cells, which are mesenchymal stem cells, CD4 positive, CD8 positive. I don't know why they don't look at the other ones. Maybe it's supplemental. But just by looking at this, you can see a dramatic reduction in the percentage of cells that have markers. These two markers of senescence, SEN-P E positive cells in these 3 cell types, and you know the flag tagged reporter in these 5 cell types, including these 3 down here. And this is acute dosage, right? So 5 days, and then there were, I believe, and then 70 to 3 days after the final dose, they were sacrificed, right? So 3 days later. So pretty impressive so far. And these are human adipose tissue explants. I don't know where these adipose tissues came from. It's probably in materials and methods, but I didn't look at the age of the donor. I didn't really check that. But again, they do the same beta-gal staining, vehicle Fisetin. Also a dramatic reduction. And I believe these are, so we're looking at various cytokines that are probably associated with senescence, right? And IL-10, is that associated with senescence? I'm not sure. But IL-6 goes down, IL-8, MCP-1, GM-CSF, colony stimulating factor, I believe. I don't know if that's significant. There's little asterisks there, but at least it didn't look as broad as a panel, but there seems to be some reduction in some of these senescence factors. Kind of the most impressive data is, like a lot of these papers, all the way at the end, which is figure 5. So I'm going to take a pause here, because here they actually do a lifespan analysis, you know, sort of the gold standard. And well, I guess since you're already looking at this, and before I take a pause, I mean, here you can see that this is a control diet, a Fisetin diet. So they have at 85 weeks of age, so they administered this. So the interesting thing is they administered this diet, the Fisetin, pretty much in mouse old age at 20 months. So male and female mice administered containing 500 mg per kg. I'm not sure exactly for how long. Well, the mice didn't really have that long to live, so it wasn't that long. And then lifespan was measured, N equals 8 to 9 mice per group. And you could see here, just by looking at this, that, you know, there's a significant increase in really for, you know, mean and maximal lifespan. And here is basically age in mice. It's the same, it's the data represented in a different format. So there is quite a bit of variance between mice, obviously, but it's, you know, it seems to be working. And these are, you know, these are what are considered normal lab mice. So these are not transgenic mice, right? So take what you will from that. I think the data up to this point is pretty solid. And based on every other paper up there, out there, you know, at the very least, you could say Fisetin is safe without any equivocation, right? It's not in this paper, but they've looked at liver toxicity profiles for mice. And I think I have the reference to it, but they have gone up to as much as 2,000 milligrams per kilogram for like 2 days for a mouse. And then they sacrificed the mice, and there was no acute toxicity, so the mouse did not drop dead, and there were no nothing aberrant in the liver. So nobody's going to be eating that much Fisetin, let's put it that way, and that's way more than is necessary for even a senolytic dose. So it appears to be a pretty safe compound by any metric, right? So that's so at the very least, and combined with all the positive data. You know I was very, not to use stock market terminology, but bearish on, you know, senolytics, because obviously things could go wrong. But that was, and the toxicity profiles of some senolytics, we looked at, you know, months and months earlier, we looked at some senolytics where, you know, the administration had to be very carefully tailored. You clearly didn't want to, you know, there's some evidence that what you were seeing was, you know, cells basically, well, let's put it this way. This was not something that, at least for some of the senolytics, the toxicity profile was not something that I want to experiment with on myself prematurely. But things like Fisetin, you know, I mean, the data is pretty robust out there. Let's see, Jeff Brakeman's. I was on a thread about Fisetin on the blog with Jessica, and we talked about bioavailability. A few self-experimenters tried it, mixing with lecithin and olive oil to boost bioavailability. Yeah, that's what I basically said. Is I don't know about these particular, you know, mixtures, but there's papers out there which look at curcumin and other compounds, you know, which are not related. But bioavailability is something, you know, that's something you really have to take into consideration. And if they haven't done bioavailability studies, I mean, these would be very straightforward to do. We're not worried about toxicity here. We're basically seeing, you know, how can we maximize the amount of Fisetin entering the bloodstream, right? And there's a lot of candidate carriers out there that are already being used, that are pretty cheap. And you could probably even start with the one they used in this paper for mice, because they used TEG and some other compounds that are non-toxic that, you know, are a bit more lipid soluble, so can solubilize, you know, Fisetin. From Steve to everyone, maybe Fisetin could be microencapsulated like the Novasol curcumin form, which showed the highest bioavailability of all supplement, more so than Norevia and ferric curcumin. Yeah. So, so definitely, you know, there's a great starting point already for known carriers out there that could be basically, you know, tried out with Fisetin, as well as any number of other plant flavonoids. Tony Vatic, is Fisetin turning off P16 gene or turning on P53 gene to send senescent cells into apoptosis? Well, it's I don't know exactly what it's doing. That's a good question. Exactly in these cells. I don't know if they're apoptosing. I know a lot of other senolytics, they've looked at so Steve, you want to chime in on this?
It really just echoes what we were saying last week about the xenomorphics versus the senolytics, and it's not entirely clear what's happening as you were working your way towards there. Quercetin, for example, is much more likely to be a senomorphic than a senolytic as originally thought. And I'm going to go out on a limb here because they're chemically very closely related. It could be just modifying the SASP bar rather than actually kills themselves. Maybe even adjusting them. We don't really know, do we? It's very hard to measure senescent cells. That's the problem.
Yeah, and
I think this is kind of an overlook of this paper of the authors. Is there are very easy assays to do for apoptosis of cells. Like you can just look at activation of the caspases, for example, and then you will really be able to see if the cells are inducing apoptosis. Of course, you also have to take into account there are other forms of cell death. There are actually quite a few number of cell death forms, but there are very good assays for all of these available.
Yeah, there definitely are. And I think there's a lot of other papers, because Fisetin has been studied by a lot of labs. A lot of other labs may have looked at, and again, does it cause apoptosis in all tissues? I don't know. That being said, I kind of like the other, the approach that these authors did was they went right to, looking at clearance of senescent cells and then seeing if the animals live longer, and they do, right? So we can back up a little bit and see in detail how these mechanisms are working as far as senomorphism or senoclearance, or but for a clinical perspective, I think the data is pretty robust that this should be tried out and as a large amount of patients as possible. Fisetin has also been shown to have pretty robust effects on animal models of Alzheimer's, for example, a number of other neurodegenerative disorders. And that's kind of what we've been harping on for a long time is when we start looking at compounds that target fundamental mechanisms that underpin aging, you're going to see across-the-board effects for a number of degenerative illnesses, which is a huge plus. So I'm super excited about this just because of its safety profile. At least in the formulations used in this paper, it's high bioavailability. Maybe not in a powdered form, but whatever, the bioavailability I think is easily breached by looking at different formulations which are already out there and being used for a number of compounds. That being said, let me go right back to the last figure that we looked at, and the only thing, if I would say that the only thing that I think is missing from here, that could be easy to do and a good follow-up, and maybe they've already done in another paper, is again more kind of physiologic behavioral measurements, right? So the mice live longer. That's great. The cells look fantastic on a petri dish. I would expect the mice to also perform better, and at least for the Alzheimer's mouse model trial that was done with Fisetin, they did have behavioral tests, and the mice did perform better. And these were tests where they basically, do these like underwater platform tests. Can the mouse remember where the platform was, right, and that sort of thing. And it certainly improved dramatically the mouse's performance. So I would expect the same performance enhancement, for the elderly mice as well, because if you're living longer, then most likely your health span is being increased here. I mean, if your mean and maximal are both going up, I would expect the mice to also, probably have better respirometry, profiles, probably better endurance, probably better, memory and other performance enhancements. Maybe that's not necessary based on all the other papers out there, right? Okay, so just to wrap it up, what they looked at. Let's look at here. I think they did the same relative expression levels of SASP in these mice as well. So these are wild type mice. I think mentioned the actual strain of mouse. Medium lifespan went up. So they did clinical chemistry on blood to measure markers of liver, so alanine, aminotransferase, alt pancreatic amylase dysfunction. So those markers went down, right? So pancreatic dysfunction, and so basically, the organs. And they looked at this composite lesion score. I'm not 100% sure. That's obviously some sort of dysfunctional measurement here. Let me just have to scroll down to how this is split to the next section. But there's lesions in various tissues, which I guess correlate with dysfunction. But basically, the bottom line is they did the same panel and profile that you've seen, and I'm not going to kind of go through that because the effects are fairly similar to what you saw before. Is that you basically have a reduced senescence-associated secretory phenotype. These are sections from kidneys which show these profiles of controlled diet, Fisetin diet in older mice that I guess this increased vascularization. I'm not a histopathologist, so this somehow indicates a more aberrant kidney, right? But basically, the bottom line is that Fisetin dramatically improved, the appearance and the functionality of tissues when it comes to reduced senescence, and factors associated with inflammation. And the bottom line is that that at least the cohort of mice that they used for this trial, 8 to 9 mice per group, male and female mixed, they lived significantly longer. I think the overall increase in mean and maximal from the get-go was about 10 to 15%, and starting at the older age, the increase was closer to 50%. So a significant increase, and again, coupled with a safety profile that's pretty much is as safe as you can possibly get. I think this is an excellent candidate to go full steam ahead with. The only thing that we need to be sure of is that it becomes bioavailable, and that's kind of a low hanging fruit kind of series of tests to kind of. I think if the safety profile is great and all this other data is great, then setting up a study to look at bioavailability followed by some aggressive studies to see if, when I say aggressive, I mean high doses, short-term acute dosages, to see if there's any beneficial effects for individuals with neurodegenerative disorders like Alzheimer's, and also people that are quote unquote normally aging. I don't see any barriers here. Again, I think the only barriers are financial, and motivational and willpower, but I don't see any scientific or clinical barriers to going ahead with, studies. Usually people have the caveat where they're like, well, we need some more animal studies. And I totally agree with this, but, at some point you got to sit back and say, okay, well, we did all the studies. It's working on everything that we have and in petri dishes and our mice, and let's if it doesn't work in humans, I would be shocked. Let's put it this way. Then it sort of invalidates every single mouse model and every single assay and everything that we know about how senescence is operating and how it's connected with aging. So I would be shocked. So what we have here, Jeff Brakeman's, also an effect that could be approved for the clearance is the cosmetic effect, which can be visually dermatologically established. Yeah, I think the effect will be more pronounced in a 70-year-old man than the 4-year-old. I'm also concerned about safety administration in regard to people above 80 or 90, because too rapid a clearance of senescent cells can be toxic and lead to a condition not very dissimilar to sepsis. Now, I'm not sure about that. I don't know where the so I know if you lyse cells, so if you lyse, so so for a cytotoxic T cell therapy, so there's CAR T therapy for cancer, and this is completely unrelated, right? So if cells are basically being targeted by natural killer cells in your body or other T cells, cytotoxic T cells, and they're kind of bursting, then you can these cells can be releasing a lot of factors where you can induce inflammation and other types of effects that are toxic to people. But I don't know if that's the case with senolytics, right? I mean, we're talking about apoptosis, which is meant to not induce, we're not talking about lysis of cells, even though there is the, it says senolytic, you would think it's not really lysis. The cells are induced to either apoptose or turn into normal cells. So I don't know if there's going to be any release of any toxins. And so I don't think there will necessarily be anything equivalent to sepsis. If I'm wrong about this, if there is a study out there that suggests senolytics can induce something similar to sepsis because it actually induces cells to lyse and kind of spew their contents into the bloodstream, please send that link. I'd like to take a look at it. I'm not aware of that. So I'm not, I mean, the only other argument I could see, against this is the thought that perhaps senescent cells have some effect that's beneficial. And, I don't know what the percentage of senescent cells there are in a 90-year-old. If your body is 90% of your cells are senescent, then I would think getting rid of all of them is not going to be good, but I doubt that very much. Okay, so I would like to see it mixed with DMSO topically. Clint Comstock does N-acetylcysteine. Clint Comstock, does N-acetylcysteine reduce the senolytic effect when taken with Fisetin? I don't know. And yes, would a topical administration to cells have some sort of effect on how your skin looks? Again, you're clearing senescent cells, so you have to realize that a lot of stuff goes wrong when you get old. You have cross-linking taking place, in your collagen. Again, what we do know is that Fisetin is extremely safe. We can most likely make it very bioavailable. So as far as I'm concerned, there's no hurdle whatsoever, ethically or clinically or scientifically, to go full steam ahead with every and all sort of trial in human subjects. So how that would work, or how much of an effect you would see when you administer it in all these different configurations, either topically or internally, or I'm not sure. So, I would expect good things, but yeah.
I would. I would. I'm going to talk about that quercetin thing again. There is a cream. I can't remember the brand, but there is a topical cream, like a beauty cream for aging. I've mentioned this before, and it has an active ingredient which is quercetin. So I'd be very interested to see a topical cream based on Fisetin, or maybe Fisetin, quercetin, and some of these other related polyphenols, mushed all into a nice cocktail. And cocktails, Peter de Keyser, who's a senolytic researcher, believes that different kinds of senescent cells use different pro-survival pathways, and that's the reason why one senolytic doesn't do them all. So I'd be very interested to see a topical cocktail applied to the skin, and you could easily do a comparison by just treating the one side of your face. Literally divide your face up and do one side but not the other, and then it sounds silly. Well, then it's a bit like that black and white guy out of Star Trek, but
then you too could be a supervillain from Batman.
You could be, you could be Two-Face, or Two-Face, isn't it?
Is that for Dick Tracy, or maybe?
But I mean, something like that would be quite easy, or you could even do it on the hand, the back of the hand, one hand untreated and the other hand not.
The other thing I was thinking is that, okay, if this is a senolytic, right? One thing they didn't look at in these mice is if it really is a senolytic and you're actually clearing cells. And here, Tony Fatica says, it would have been interesting if the paper showed any reduction in visceral fat from senescent cell reduction. And that's I was just thinking that when I was reading the paper. I recall Judy Campisi saying most senescent cells are packed in visceral fat. Maybe that's why exercise, CR, and consumption of fruits, vegetables have the same effects on visceral fat. So yeah, Tony, I think that's an interesting thing to look at. If if you take a lot of senolytics and it's actually clearing them and they happen to be in visceral fat, will you also, as a positive side effect, lose weight, right? I don't know.
And we could use MRI to have a look at visceral fat.
Yeah. So there's a lot of things that could be followed up, to me, even more excitingly, directly in humans, right? That's why I'm excited about this paper. It builds on a mountain, a literal mountain, or maybe a large hill of published research out there that's looked at Fisetin, and other safe senolytics, flavonoids, I think mostly. And yes, the dosages have to be very high, but the toxicity is pretty much negligible, so that's irrelevant. Number two, these compounds are cheap. I mean, if you go online and you look at how much they go for, even from chemical supply houses that sell pure ones, it can get a little pricey, but we're not talking about the price of antibodies here. These are pretty inexpensive compounds, and the other thing is, you don't have to be taking this if the mechanism is what we've talked about. We're talking about an acute dose for a very short period of time. So you don't have to be taking this chronically, starting in your 20s for the rest of your life. We're talking about relatively high dose for a short period of time to initiate either a senomorphic or some sort of senotherapeutic effect. So I think to me, all of the everything's in place, to basically start setting up the right trials in people. And another comment, Jeff Brakeman's, I think it makes sense to senolyticize the skin. It's the largest organ in the body, one of the first lines of defense for the immune system, and easily reachable. So senolyticized skin would have a small pan-systemic effect too. Yes, Sven, not if you buy it from Sigma, but also, Sven, if you buy water from Sigma, you'll also go broke. Also sugar and salt, NaCl, pretty much anything if you buy from Sigma will be its expense translated into the weight. It's equivalent weight in platinum and gold.
I looked, it's 60 euro for 100 milligram.
It's a joke. But I mean, I was going to just to circle back what we were talking about dosages. By reducing the amount of dosage that you'd need would be to actually make the bioavailability higher. So it's probably a relatively high dose because in general the flavonoids tend to be pretty poor bioavailability. Certainly with quercetin, apigenin, I'm not sure about curcumin, but I think possibly. But again, there are ways and means around that. And as you said earlier, there are different delivery methods. So again, that is something else that could drive the dosage and therefore the cost down as well.
Yeah, I mean, let's look, and we really, I mean, the cost definitely will go down, but we're already talking at a remarkably low cost. I mean, compared to a chemotherapeutic antibody that's one dose is probably $250,000, and if you're lucky gives you a few extra months of life, and we're talking about something here that could potentially do a hell of a lot more than that, and is basically pretty much as cheap as it can go. Maybe it can go cheaper, but it's pretty much already within the reach of the majority of people. So one thing we talk about here, and I mean, I don't know, is it too good to be true? I hope not. I'm super optimistic because this has been repeated by a lot of different independent labs, and I'm a biologist. I believe in evolution, right? I believe that everything is evolutionarily related. That's Dobzhansky said it best when he said nothing in biology makes sense except through the lens of evolution. I'm paraphrasing that, but that's pretty much correct. So if you see this kind of robust translatability, this between different organisms, you know, and compounds like Fisetin, certainly we've seen that with CR, then it's, I would be stunned if you wouldn't see any beneficial effects in humans, a significant effect. It would, you know, so for that reason, what I'm really more stunned about is that we don't have, something must be dramatically wrong with our framework of testing. If me clicking on the U.S. trials database and I don't see 200 or 300 Fisetin trials. Go on it right now. You'll probably find 2, maybe 3, and both of them are using like small dosages, not even looking at bioavailability. I think small dosages in some sort of pill form, right?
Can't be patented. Follow the money,
yeah,
you know. I mean, at the end of the day,
but you can, we can be cynical about it, but you can patent formulations. You can definitely.
And you can also modify the molecules. That the experiment we were talking about last month with the curcumin and the Fisetin, they actually also, if you recall, they made 3 derivative molecules based on them, and it was J147 CA.
Yeah.
Not very memorable, whatever it was, but they can patent those because they're modified forms and more stable.
Yeah, but regardless,
they already mentioned in this paper at the end that they have made variants of Fisetin that are better, that have a higher senolytic effect, but they don't specify anything there.
Yeah, well, I'm impressed by what I've seen already thus far with this compound. I think if you can boost the bioavailability, and I think I'm gonna kind of shoot from the hip here and say I'm impressed with what I see from this compound already. I think the bioavailability can be improved based on what's already out there, published off the shelf. There's dozens of different formulations that are already being applied to, you know, other similar structure molecules that are known to increase bioavailability. So basically finding the right one should be pretty easy. And I think going ahead and designing these trials should be #1 top priority for the new year. I think it would be a shame, you know, come Journal Club 2020, and we are still talking about animal trials with Fisetin and other compounds. I think it would make no sense to me. I don't know. Somebody who's in the industry can correct me, but I think it's already kind of. I'm convinced.
No, I think it will happen. There's growing to get this sort of thing done, and we already know that we even know that just the regular Fisetin works and curcumin, because that paper we did last time, it blatantly says, this stuff works, but you know, we have obviously got some better molecules, but it does absolutely work, and it reduces the presence of tangles. So if that's not impressive, I don't know what is.
And Jeff Brakeman's just sent out a question. What about co-administration of Fisetin with other senolytics that he mentions here? You know, a lot of these compounds probably have cross-reactivity in multiple pathways, and just like a paper that we looked at, I believe a couple of months ago, which was I don't know if it was just senolytics. It was a mouse model looking at a computational way to kind of look at substances that can synergize, right? I can't recall which ones they looked at. It might have been quercetin with some other compounds. I know you remember the paper. I'm vague on it right now, but basically, I think, you know, I think there's a lot to be said with co-administration of compounds that have a lot of cross-reactivities, and you know, getting this, you know, quote unquote synergistic effect. And Jeff Brakeman's again, I heard that Life Extension Foundation also jumped on the bandwagon with their senolytic supplement combining quercetin phytosome theaflavin from black tea? Well, yes. I'm sure they have. I'm sure a lot of, if you go to Amazon right now and just punch in Fisetin, you just get a long laundry list of bottles out there. Again, my issue is it's available, it's safe, available on the market, but is how much should you be taking to get an optimal effect? I'm not worried about the toxicity per se, but I would be worried about is the quality control there for the company to make sure you're actually getting the dose of Fisetin you're supposed to be getting? How much Fisetin should you be taking? Because you really want to hit that optimal dose to initiate senescence clearance. You don't want to be underdosing. I don't think overdosing is a problem in this case. What else is there? Oh, and bioavailability. So let's say you based on your research say, I got to take a gram of this stuff, but how much of it is actually getting into your bloodstream? Is a pill form using a dry formulation the best way to get it into your system? I would go out on a limb here and say probably not. That's not how they administered this to the mice, right? They administered it either mixed with food or in a slurry that was basically, I can pull up the paper here, it's in front of me, but basically it was their own kind of, their own sort of oralness. Oh, you can look up these compounds, but it's in paper. 60% fosol, 50 PG, 30% PEG 400, and 10% ethanol, right? So it was basically there to solubilize the compound, right? So these compounds have different solubilities, and you want it to be dispersed and soluble so it can actually penetrate cell membranes. And there's a lot of studies out there that have shown that taking something like curcumin in a pill form versus mixed with food, mixing it with something that's going to solubilize so mixing it with food that's kind of fatty, it's probably a better way to get it into your bloodstream than just taking powdered curcumin. So those are the issues that I would have with making sure that I actually got a therapeutic, or what I would consider to be a human therapeutic dose. And maybe between now and our fireside chat, might be able to have time to pull up a paper that maybe if they've done some more thorough pharmacokinetic studies, and we can talk a little bit. I can talk a little bit about that in a more educated manner.
As long as we're allowed to talk science, of course, on the fireside. The fireside Christmas chat on the 22nd. We'll be announcing the details soon, but as long as we don't get told off by Elena for talking too much science, Oliver. The other thing I wanted to add to that list of things that you brought up was the other thing that's a concern is interactions. So, how do we know that this flavonoid doesn't interact negatively with this?
We don't.
We don't really, and I think we need to do more fundamental tests before really I could confidently say, yes, you should you should be all right with this particular combination or that particular combination. And I think it needs to be done. I mean, which is one of the things that we're doing with the MMTP actually is we are testing multiple combinations of senolytics right now to try and get some of those answers. So,
Sven suggests a nice alcoholic cocktail for the holidays, not to promote alcoholism, but 10% ethanol and Fisetin and a bit of PEG. I would say just dump it in your eggnog, but that's me. You've got lipids in your eggnog, you've got alcohol. I think eggnog might be the best carrier.
I don't know about that. We don't really get it here so much. Although I think some of the supermarkets nowadays do tend to stock it even in the UK, but it's not really. And my wife's looking at me like I'm crazy, but eggnog's not really the sort of thing that we would traditionally drink here in the UK at Christmas. Really? I think the closest. Yeah, I know you can get it in like cartons and things, can't you? And I think I've seen it here recently as well. The sort of eggy thing that we drink at Christmas traditionally, and I'm showing my age here, is Dutch Advocacy, which is what I call alcoholic custard. if anybody's ever tried it. Advocacy.
Well, it's basically what eggnog is, right? Sort of like a thin kind of custardy, like.
I'd probably enjoy it.
Yeah. So yeah. So again, we're not advocating self-experimentation here. People are coming in with comments about this is what I've tried. Good idea. I'm not sure. I don't know. It's I can't comment until I have something in front of me that says, okay, this amount of dosage and mixed with this carrier is going to give you this amount of micromolar Fisetin in your bloodstream, right? And we're not, and we're still not talking, and we're just saying bloodstream, right? We I don't know how much of it actually gets through the blood-brain barrier, for example, but I think all the ducks are lined up to basically do some really breakthrough experiments in humans. I think I think the safety profile is as perfect as you can possibly get for anything that's potentially testable. I think the price is right for this stuff, this compound. So I think it's just a matter of getting the right participants, the volunteers, the protocols, and making this go. And that's something that I'm, not to toot my own horn a little bit, but that's something that I'm kind of working behind the scenes to come up with a list of, here at Leaf, a list of ultra safe therapies, potential therapies, Fisetin being one of them, that could be immediately translated into a variety of different clinical trials, either crowdsourced or otherwise, that should be, that we can go full steam ahead on, because A, they've been replicated, up the wazoo by every lab that we can, every reputable lab. The safety profile is outstanding, meaning I would not feel any risk whatsoever, administering it to myself. And the cost is relatively low. So really the only barrier is getting the participants together and getting the protocol together, and kind of making sure that we get statistically valid data. That's really the only barrier. And there are yeah, none of that's easy, but really all the hard work, I think the hard work has been done, which is figuring out the best candidates, and Fisetin's one of them, and there's other stuff out there that that as I've been kind of pouring through all the papers, including the ones that we've read in Presented Journal Club, you start to think, okay, well, we sort of have a candidate list of stuff that basically could be tested out, and based on everything that's published, with very little risk of toxicity and also failure, right? Because that's you don't want to waste your time, especially when you finally get down to humans. You're not using humans as your initial screen here. But I think for a lot of these compounds, we're beyond that point. And I think hopefully in the new year, we'll see that off and running, and we'll see some fantastic results. That's my New Year's wish.
It's a pretty good one. Apigenin, that's my pet inside tip. I think I'm pretty confident about apigenin. I can send you some papers about that, Oliver, which relate to NAD+.
Yeah,
and
please
how
do.
And how quercetin, there you go, quercetin again, and apigenin both appear to restore NAD+ levels, presumably by inhibiting SASP. So I do wonder what effect curcumin and Fisetin might have on NAD levels within the cell. Again, that's something, that is another thing that we could look at here as well. Lots of things we could do. Maybe we'll get a top 10 list together and start actually getting stuck in and doing some of those trials. It's certainly easier nowadays to collect data.
Yeah.
you know, with automation and
it's easier to buy stuff. It's easier to collect data. It's easier to talk to people. It's easier to communicate. I mean, the infrastructure is there. I mean,
yeah, Apple do an application now. I think it's called Lab or Lab Kit, and it essentially allows you to run clinical trials, and then all the person needs is an iPhone device, and then they can actually remotely submit data, and it collects it all. So, you know, enrolling people in clinical trials isn't the major undertaking it used to be. I mean, even AgeMe was fundraised for, has that kind of facility. And again, you could use the age meter we could even use the age meter if we organised a little trial, because you're going to notice potentially functional aging changes, maybe, if it works. I think we should do it. And as you say, I think if it's 2020 and we're still talking about Fisetin animal trials, I think something's gone badly wrong. So
yeah,
I'm with you on that Christmas wish, I think.
All right. Well, there's no further questions. I wish everybody a Merry Christmas, belated Happy Hanukkah, Happy Kwanzaa, Happy Holidays, Happy whatever you're celebrating, Happy Pagan New Year. Or if you're just not celebrating at all. But if not, then shame on you. Find something to celebrate.
Well, being alive and healthy is something to be glad of. So we'll have more of that, and hopefully, Senolytics will help more people enjoy that for longer as well. So, you know, live long and prosper, as Buck would have said. And I think that's a good. I think that's a good thing to live by. So that's great. I think that's about it. It's been interesting. We've had a lot of questions actually this week, which is fantastic. We do like people getting involved and joining in. So
I figured just because this paper was the most translational to some, you know, to had the shortest gap to translating to, you know, human effects and trials. So that we've read thus far.
You know, these things, these sorts of things are very accessible and very applicable to people right now, and with a bit of know-how, they're easily actionable. A lot of the people in the audience seem to be biohackers, and these people, you know, they've got the means, you know. So it is obviously not something that we're suggesting you try at home, but if you know what you're doing, then you know what you're doing, and that's all we can say really. So thanks for joining us anyway, and I'm not sure what the topic is going to be for next time, but we'll let you know for the usual channels. And if you did enjoy the stream, check in with us next time. And we actually run this stream thanks to the support of our patrons, the Lifespan Heroes. So if you did enjoy this and you're interested in supporting ongoing streams and activities and helping us out, if you would maybe consider checking out Lifespan Heroes at www.lifespan.io/hero. And that's about it. We'll see you next time, and stay safe, and hopefully everybody have a happy holiday, and we'll see you next year.
All right. Adios.
Happy holidays. Bye-bye.